Cutaneous melanoma is the most common category of skin cancer in the U.S. and also the most deadly. In 2023, it's expected that about 97,610 new melanomas will be diagnosed, and an estimated 7,990 people are expected to pass away from having it. The risk of being diagnosed with melanoma is higher in males due to phenotypical reasons such as ethnicity, eye color, hair color, as well as environmental and behavioral reasons such as their occupation or lack of sunscreen use. These factors put them at high risk for diagnosis over females.
Intro and background continued. A functional TP53 tumor suppressor gene codes for a DNA binding protein that is involved in cell death. The mutation of this gene causes immortality of the cell. It is known that a cytosine to thymine tandem mutation is the marker for UV-induced gene alteration. That mutation is a sign of extensive sun exposure. These same mutations are found in TP53 tumor suppressor genes located in cancer and sun-exposed areas, meaning the presence of TP53 mutation is a good indication of UV exposure. Mutation of TP53 is located in 90% of squamous cell carcinoma and 50% of basal cell carcinoma. No hypothesis. No statistically significant association exists between melanoma development in males and females regarding UV sunlight genetic mutations, gene expressions, or the resulting multifaceted phenotype. Alternative hypothesis is that statistically significant association existed between melanoma developments in males and females regarding UV sunlight genetic mutations, gene expressions, or the resulting multifaceted phenotype.
This part is the method. So for the method, we use the GEO2R to discover the genetics, the phenotypes, and the expression, as well as the polymorphism data. Also, for male and female, we use the National Cancer Institute GDC data portal for that. And including this, we'll assess in any uncovered variant that is associated with a trait. And also, we will use we will base on the method to contribute to discussion and conclusion.
The main focus was on analyzing the TP53 gene using data sourced from the National Cancer Institute and analyzing it using an omics tool known as GeoR2. Our analysis revealed a significant correlation between TP53 expression and melanoma development, specifically in male patients. This gene was found to be highly expressed in male cancer cases, thus suggesting the link. Also, the rate in males was higher than in females, and thus was also accompanied by a lower survival rate. We explored a numerous of differential gene expression by examining cells treated with mock conditions versus untreated cells, and also treated with UV radiation and those treated with adrenaline. Our findings suggest that we identified several sets of differently expressed genes when comparing untreated cells to those treated with UV. This suggests that UV treatment significantly influences the gene's expression compared to both the untreated and the adenosine treatment cells.
As the data shows, there is a high rate of mutations of TP53 and significant expressed genes in those who have been exposed to UV rays from the sun. We found that there is a confirmed statistically proven association between UV exposure and cancer in humans. 90% of TP53 mutation in melanoma was GT2AC and does serve as initiation of the process to start the cells toward the development of cancer. Atreomycin is a cancer treatment medication that reduces the expression of TP53 genes significantly based on the data we provided. However, it does deliver the toxic side effects based on the research. And one of the research studies shows that atreomycin loaded with exosome can progressively exhibit the targeting cancer cells and reduce the migration. There's also significant difference in the gene expression in males who are exposed to UV over females in the same scenario. And testosterone has found to be one of the possible factors that cause increase of melanoma. in male. Additionally, males tend to use less protective sunscreen than female, therefore contribute to the reason why male has higher rate of melanoma.